All right, this is the lecture for chem477. I was trying to finish this yesterday, but it just took too long. So here's the lecture. This is actually this is in the slide deck that you guys have on the iLearn site. So I changed it and I'm updating it with some more stuff. Okay, but you saw some of this already. Let me see. Okay, so we saw this already. PGP was the multi-drug resistance pump, and it was in the last lecture. It was a mechanism that a protein, a transporter, is a transporter protein that kind of kicks out small molecule drugs from different tissues and stuff. And it was kind of how loperamide, the anti-diarrhea drug, which is a mu-opioid agonist, how it acted as an anti-diarrhea drug and doesn't have any CNS effects really, like the way the other mu-opioid agonists do, like morphine or fentanyl and things like that. Okay, all right. Anyway, and we also showed the X-ray crystal structure of this. We talked about it and how small molecule drugs kind of get effluxed out, exported. This is a pretty crazy protein, but it's a transport pump, and that's how it works. That's the X-ray crystal structure. We talked about that's the PDB file. This might be something you can open with the IC3ND, but it's also a little bit of a big protein. You can try certainly. I use a program called Pymol, which is very cool, and you can try to figure it out yourself. It's a little hard for me to explain how to install it and stuff, but I do have PC program installation programs, but it's still a couple steps. But this is Pymol, the program that generates this, right? Okay, so kind of going on from where we were, we last time were on the whiteboard we talked about some definitions. We talked about an introduction to pharmacodynamics and kind of how drugs have effects in body. Intro to pharmacokinetics. Really, the four things were the ADME, absorption, distribution, metabolism, and excretion. We talked a little bit about clinical pharmacology and sort of the steps that you take to get drugs on the market. And we also talked about toxicology a bit. And toxicology is sort of a related field to pharmacology. Okay. So what are we going to do next? And try to get through today. And I don't know if we'll get through everything, but a little bit more on drug names and nomenclature. We started that. I think we're just sort of finishing that up in the last whiteboard lecture. Pharmacodynamics, a little bit more detail on pharmacodynamics. So we started it. Let's talk a little bit more about kind of how drugs interact with a biological organism. Dose response, we'll get into a little bit more in dose response. And show different types of agonists and antagonists. And then we'll go from there and show kind of the dose response that you expect with different agonists and antagonists. All right, cool. So we showed this molecule, and this of course is aspirin, as an example of drug names. And let's just repeat this real quickly in this PowerPoint format. The IUPAC name is 2-S-toxybenzoic acid. So of course benzoic acid is sort of the top part, and then acetoxy kind of refers to this piece. IUPAC is a very formal naming system. And there's also a chemical name or a common name, which is acetyl salicylic acid. Sometimes people abbreviate that as ASA. And that's a derivative of salicylic acid, with salicylic acid, which is this. So the salicylic acid is this molecule. This is actually used also for some pharmaceutical purposes, not as a NSAID like this, but I think it's like maybe a anti-wart cream or it's in some anti-wart medicines. Acetyl salicylic acid. What does acetyl mean? it means that, well, you have an acetyl group, and the acetyl group is on this oxygen. That's what we say over here. It's a derivative of salicylic acid. Generic name is aspirin, non-proprietary pharmaceutical name. So this is the name that you're familiar with, but it also has trade names. Some of the trade names are Bufferin, Ecotrin, Aspirin, and these are proprietary company names. And of course, Bufferin that you might be able to buy at the store is one of the examples. All right. Another example, of course, this is Tylenol, and let's go through the naming of Tylenol. The IUPAC name is N-hydroxyphenyl acetamide, and so 4-hydroxyphenyl, of course, is the hydroxy and the benzene, right? And then the acetamide refers to, well, actually, the way this IUPAC name is constructed, it's 4-hydroxyphenyl is something that's on the nitrogen. Okay, so of acetamide. So acetamide is really just this piece, right? That's acetamide. Like acetic acid, but there's the amide version of that. And 4-hydroxyphenyl is this thing, and it's kind of like something that is on the nitrogen. So that's kind of how the IUPAC naming of this goes. And IUPAC's very formal and has very this is very rule-based, right? We don't use the common names in IUPAC. So what's a common name version of this? And you don't have to know this. I'm trying to review your organic chemistry and kind of the silly nuances of chemical naming. I'm not going to test you, or this wouldn't be. I may. We'll see how the quiz or the homeworks go, but you will see kind of names in different formats. Chemical name of this is N-acetyl para-aminophenol, and common naming again is different than IUPAC naming, and phenol, first of all. Phenol. What's phenol? Phenol is essentially just the benzene with an alcohol on it. That is not IUPAC. That's what we call a common name. And so aminophenol would be essentially this molecule. This is aminophenol. And so Tylenol is a derivative of aminophenol. And we would say it's N-acetyl para-aminophenol. So you can see if this is para-aminophenol, that N-acetyl would just put the acetyl group on the nitrogen. So this just shows sort of the nuances of naming that kind of molecule also. All right. Generic name, acetaminophen or paracetamol. These are like the generic non-proprietary names for Tylenol. Okay. Acetaminophen, I'm sure you've heard of. Okay. And then the trade name is Tylenol, which is a registered trademark by the company Tylenol. Okay? Another one. Okay, so so this is fluorouracil or 5-fluorouracil. We saw this as a anti-cancer drug. It's a kind of like a nucleotide base analog. It gets incorporated into the growing DNA of tumors, and it kind of messes it up and it kills the cells. So it's a cytotoxic agent. It's a chemotherapeutic drug that would be used if you have cancer. Okay. Chemical name is 5-fluorouracil, IUPAC. So the IUPAC may actually be a little more complicated because I don't know if uracil is recognized in IUPAC. So there may be even a more complicated name, but Uuracil is pretty well understood as like this molecule minus the F. Okay. And then the chemical name, common name is basically the same thing. There's not like variations is the point I'm trying to say. And also the generic name is 5-fluorouracil or even maybe just fluorouracil. That's kind of understood as the non-proprietary name. But as a trade name, as a drug, it's called Uuracil. And that's a proprietary drug. This does not look like a solid. It looks like a injection. It's a IV type drug you would get in chemotherapy. Okay, to prevent cancer growth. What about this lovely molecule? Well, this is Taxol. I don't know if you've seen it before. It's a pretty complicated molecule. It's isolated from the yew tree, and it's a natural product, a naturally occurring anti-cancer drug. It inhibits like beta-tubulin polymerization. It is a great cancer drug, and it's clinically used. So, chemical name IUPAC, this is beyond the level of IUPAC. So, and common naming, this just doesn't make sense. With a molecule of this complexity, you can't really apply these kind of naming systems, and a lot of compounds like that with complicated structures. Generic name is Paclitaxel. So this is the name that it refers to the drug as a non-proprietary entity, and like a generic name, yeah. And then the trade name is Taxol, which is proprietary. And it's also like an IV use with a septum on it. You stick your needle in, suck it up, and it would be administered IV. Okay? All right. Moving on. Pharmacodynamics, a little more detail. What makes a molecule a drug? Think about that a bit. What makes a molecule a drug? What makes a molecule a drug? I mean, we kind of know what drugs are. We kind of know what molecules are. And what would make a molecule a drug? And how might you define a drug? That's kind of something you may have not ever done before. It's like, how do I define this concept of a drug? Well, one definition, and this is again, we're following the PDF, the text-based PDF of pharmacology. So a lot of this is kind of taken from there. It's a pretty nice little summary. Anyway, one definition of a drug is a chemical that perturbs the biological system for a therapeutic effect, right? Makes sense. We are perturbing a biological system, a physiological system, to get a therapeutic effect. And that would be useful. What would be a toxin? A toxin might be a chemical that perturbs a biological system for a toxic effect or negative effect, which is usually not something you would want to take. Possible drug targets are proteins or DNA, but is that always the case? I mean, when you think about a drug target, you might think, oh, well, the drug binds to protein and causes some kind of downstream effect, or the drug binds to DNA and causes some downstream effect. Is that always the case? And the answer is yes, but not always. There's a couple exceptions. A couple that I found, and just kind of thinking about drugs, is exceptions, of course. Antacids. That is a drug you would take, it's a pretty simple pharmaceutical, calcium carbonate, and calcium carbonate would kind of treat heartburn or gastroesophageal reflux disorder, sort of like those kind of things, and pretty effective drug. It's not really interacting with a protein or a DNA. It's really interacting with acid in your stomach, right? What's another one? Another one that I I've thought about is a monochloroacetic acid. Monochloroacetic acid. It's, of course, is acetic acid with a chloro, and it's not dichloro or trichloroacetic acid. It's monochloroacetic acid. This is actually used as a drug to treat plantar warts, which are human papillomavirus. I've actually had this one time, a long time ago, probably 10 years ago, from going to the gym, and I think I took my flip-flops off for a second and got it at the gym. So when you're at the gym, you should wear your flip-flops, right? So there are chemical or pharmaceuticals like Uuracil, which is monochloroacetic acid. That's what a plantar wart looks like. It's not fun. It's really hard to walk if you have a plantar wart. What does it do? It's not interacting with proteins and DNA in that kind of traditional sense, but you kind of apply it locally, and it really just eats all of the wart material and the papillomavirus, which is kind of like a growing entity in your foot, and it sort of destroys it. It's just using it as a super strong acid. So you can almost imagine sulfuric acid or something, but that might have some secondary effects to do the same thing. I think they also use like liquid nitrogen to sort of freeze out the wart. Why does it work? Well, pK is 2.8, and that's not quite the sulfuric acid. It's more acidic than acetic acid. It's more acidic than acetic acid, but it's not like super acidic like sulfuric acid. So this might be like a sweet spot of acidity. to kind of do the damage to the HPV, but not to actually like destroy your foot. And you are applying it locally, but still you have to, you know, it's, I think they fine-tuned this and found the right pKa value. Of course, if we look at acetic acid, the pKa is 4.7, so this is not as acidic, and this probably wouldn't be as effective as a treatment. Otherwise, people would use it. I'm sure some people try to use it, but it's not, you know, acetic acid is not very biologically destructive the way this might be. Okay. All right. I'm gonna switch videos. Stop recording. Okay, I should be back. I am back, I hope. I'm recording again. Okay, so let's now talk about protein receptors or DNA and kind of as drug targets, because most drugs are targeting protein receptors or DNA. And so what are some of the types of proteins, and that are commonly targeted by drugs, with the drugs being agonists or antagonists? One of them, and I don't know if the text is easy to read or not, but you can also remember the PowerPoint should be online too. Ligand-gated ion channels. Ligand-gated ion channels. What does that mean? Well, your drug is a ligand, and it binds to a receptor on the outside of the cell, and it causes, and it's an ion channel, so it causes ions to flow through the protein, and that'll cause some downstream effects, membrane potential or ionic concentration. It may trigger a synaptic response, like in a neuron or something like that. An example is the cholinergic nicotinic receptor, and that's kind of how nicotine works and has its sort of stimulant activity. Another class of these of protein drug targets are called G-protein coupled receptors. G-protein coupled receptors. Example are alpha and beta adrenoreceptors, which are like adrenaline, right? And we're familiar with adrenaline as a, it's a hormone that we use in our body that causes us to get like a fight or flight response. It increases your blood pressure, your heart rate, and things like that. And anyways, another class of drug targets are GPCRs. They're called GPCRs, G-protein coupled receptor, GPCR. So what happens is a drug binds on the outside of the cell to the receptor. It causes some sort of conformational shift. It's not like ions flowing through, but it's more like a conformational effect. And that causes some kind of protein phosphorylation and then some kind of downstream effects, intracellular effects. Okay. What about an enzyme-linked receptor? So the insulin receptor works this way, where insulin binds on the outside of the cell, it causes some effect, and then it's like something happens in the inside. So it's like you're just like a kinase or something, a kinase, which is a protein that phosphorylates things. That'll become active, and then some sort of receptor phosphorylation, protein phosphorylation, and that has some downstream effects like within the cell. So insulin does this. There's a class of kinases called receptor tyrosine kinases, receptor tyrosine kinase, RTK, that do the same thing. There's a bunch of these that are drug targets for cancer. If you actually Google enzyme-linked receptors, or even just go to the Wikipedia page, you'll see a whole bunch of these kind of kinase-type drug targets that are enzyme-linked receptors. Same with GPCRs. So GPCRs are a huge class of drug targets that the pharmaceutical industry uses. Intracellular receptors, like the steroid receptor, and just a lot of things. Where the drug, it just gets through the membrane. It has to be kind of greasy, like a steroid. It gets through the membrane, and then it causes some kind of effect. It binds to like a protein that's maybe in solution, like a cytoplasmic solution protein, and then that causes intracellular effects. So these are kind of a few of the ways where proteins are used as drug targets. Okay. Other drugs bind to DNA or even RNA. There's something called RNAi therapeutics. RNAi is I'm not going to really, at least for now, get into RNAi, but it's a type of way to sort of shut down protein expression. It's kind of related to the new CRISPR-Cas9 technology. But anyway, there are drugs that bind to DNA, cancer drugs, and also maybe RNAi therapeutics, which interact with RNA and kind of sort of silence genes. We call it gene silencing. And here's an example of a normal piece of DNA. And then what are these little black things? Well, they are drugs that are stuck into the alpha helix. What's the name of that? It's called DNA intercalation. I'll show that word in a second. Intercalation. DNA intercalation. Here's an example of DNA intercalation. So this is like the alpha helix of DNA. And this is a molecule called ethidium bromide, which is not a drug because it's actually very toxic. It intercalates DNA, so the molecule, it's very planar and aromatic. It slips into the alpha helix. And it's usually as a dye. It's like a dye or a stain for DNA gel electrophoresis. So if you're in a biochemistry lab and you're purifying DNA fragments, you use this as a dye to dye your DNA and then isolate your little pieces of DNA. So it's more of a tool that we use in biology or biochemistry. Okay. Here's another one. This is a crazy looking natural product, D-actinomycin. That is the generic name, D-actinomycin, and the trademark name is dactinomycin. So they just took the D, got rid of the space, and now it's called dactinomycin. It's a chemotherapeutic DNA intercalator. So this is used in cancer as another cancer chemotherapy. So it's also a liquid. You inject it. It's an IV. So intravenous solution. So this avoids oral breakdown, and it kind of gets right into your system, and it's kind of needed for this sort of drug. Okay. All right. So let's look at a couple of these drug target types by X-ray crystallography, because now we're getting comfortable with X-ray crystal structures. A lot of these examples actually have X-ray crystal structures. That's really cool. So let's look at a couple. So remember some of these. We have ligand-gated ion channels. Ligand-gated ion channels. We'll show one of those. We'll show a GPCR. In this case, I didn't have a good example of an enzyme-linked receptor, just because I couldn't find a crystal structure that has the whole thing. Like you can get an X-ray crystal structure of the insulin receptor, but it's usually like the outside part and not the inside part. So that shows some of the limitation of crystal structures. Also intracellular receptors, which basically means any protein on the inside of the cell. And I'll show a couple of those sort of things too. Okay, ligand-gated ion channel. So this is the cholinergic nicotine receptor in complex with the drug nicotine, which is not usually a therapeutic agent. It has biological effects, but I mean in most cases nicotine is used while it's part of like tobacco or something. Well, it's a nice crystal structure. Look at that. This is a high resolution X-ray crystal structure, a pretty crazy molecule. So it's a membrane embedded protein. So you have all these alpha helices, and what happens in this case, you have sort of the extracellular domain and the cytoplasmic domain, this usually resides in muscle cells and also neurons, and it kind of has peripheral effects and neurological effects. What happens is the red, which is nicotine, it binds to this receptor. The drug binds to the receptor, and the structure is a pentamer. So it actually has five proteins that are all complex together. They're the different colors. You have yellow, sort of tan, purple, green, and cyan, and there's maybe something in the back too. But it's a pentamer, and it's a ligand-gated ion channel, and so it allows ions to flow through. So when nicotine gets bound, and it's an agonist, what happens? And there's nicotine, the little red spherical thing. it allows sodium to pretty much flow into the cell and potassium to flow out, and that has all the kind of downstream effects that cause the effects of nicotine that we're used to observing. PDB code is 6PV7. It's visualized in Pymol, and you could actually probably open this up with that tool that I gave you. It may be kind of complicated, but you can certainly give it a shot. The membrane position is roughly here. It's in the alpha helical region. Okay. Could you imagine other drugs that have the same effect? And yes, there are, including, I mean, this is a cholinergic nicotine receptor. And there's other, it's actually, I think it's a nicotinic cholinergic receptor. I probably have to double check that. Yeah, I think that's actually, let me check that real quick. Get it right here. Yeah, the names may be wrong up here. It was called this the nicotinic cholinergic receptor is kind of the proper name. But anyway, there's other agonists, and some of them are related to choline or acetylcholine. So acetylcholine does the same kind of thing. It's an agonist, and it has the same kind of effect. So this is a naturally occurring substance in our body, kind of the natural ligand, natural agonist. Here's a weird molecule. It's called epibetidine. It causes numbness, paralysis, and possibly death. So it's not something you'd probably want to take therapeutically, but it's actually through the effects of this and kind of like a high, since it's potent, it can actually kill you. And it's actually found in this poison arrow frog, which is part of how people would create poison arrows in the wild, is kind of getting substances off a frog that have some sort of toxic effect. So in this case, this is actually a toxic substance that's used sort of as a kind of a poison for poison arrows, right? So that's an interesting little twist. Okay, G protein coupled receptors. The beta adrenergic receptor in complex with an antagonist, which is carazolol. Carazolol is a beta blocker. It's one of these blockers. Okay, so what does this look like? In GPCR, remember, it was like a protein that's embedded that has a kind of an intracellular response, phosphorylation event, and a cascade of events. So this is what it looks like. There's the membrane. It's an embedded structure. It's a membrane embedded protein. There's an extracellular side, a cytoplasmic side, and this is actually found in the heart. So this is the interface between the outside and inside of the heart. There's the drug. What actually happens is when an agonist would bind. This happens to be an antagonist. When an agonist would bind, it would bind to the outside, and then you'd have. Actually, let's go back. GPCRs are found. They're common drug targets. About 34% of all FDA-approved drugs target about 108 different GPCRs. So they are common drug targets. That's what I wanted to say. Okay, and so what happens though when an agonist would bind? It would cause some sort of a change in the structure. It's crazy that it does that, but the alpha helical arrangement changes in a way that causes G protein phosphorylation, which are just some downstream proteins, and that leads to, in this case, increased heart rate, blood pressure, etc. So that's kind of how it operates as its sort of native functional protein, right? And so when we add this stuff, carazolol, which is a structure, it's a beta adrenergic antagonist, or we call it a beta blocker. It reduces blood pressure and heart rate because it binds in a way that kind of blocks the receptor but does not have the downstream effects. And so it ends up, your net effect is decrease in blood pressure and heart rate, which is what you want, right? In a lot of cases. Like, how would this be used therapeutically? Like, if you have high blood pressure, if you have hypertension, if you have diseases that kind of, well, hypertension is a big source of heart disease and other problems in the world. So people would take beta blockers. People often frequently take beta blockers to treat those sort of conditions. Okay, this was PDB5X7D. You can also download it and visualize it. I use Pymol, which again is my kind of cool program that I use to do visualization. All right. Indinavir. Indinavir is an early HIV-1 protease inhibitor. So this is kind of this is a little different because it's not a membrane-bound protein. This is part of HIV, right? And so you get infected with HIV. What did it do? What was the what did it do? I have a little mini slide which kind of reviews the basic idea of HIV and how viral mRNA gets translated into proteins, and you just get this crazy big protein sequence, and then and HIV is sneaky in that it creates one big piece of protein that encodes all of its proteins, and then it has HIV protease, which is the little scissors that snip the proteins into little mini pieces. These are all the proteins that HIV uses: the matrix, the capsid, which is involved in the viral, outside of the virus, nucleocapsid, reverse transcriptase, so it creates. That's a drug target too, remember? HIV reverse transcriptase. It makes its own protease. It makes various proteins that are involved. Integrase we talked about also. All these proteins are involved in HIV and required for HIV to be a functional virus. And isn't that crazy? It's like I'm still blown away by how this works, and like it seems it's like a very almost intelligently intelligent system. It doesn't really make too much sense. Well, I guess it makes sense. I mean, it evolved, but it seems like very logical, right? The way this works. Anyway, so what does HIV protease look like, and how does it look like as a receptor? Well, this is a kind of a quickie overview. Of course, the drug sits in the middle. So I always say that the protease inhibitor blocks the scissors, because the protease is a pair of scissors, and this whole thing is a pair of scissors, and this is jamming the scissors. So it binds in a way, indinavir binds in a way that prevents the scissors from operating. This is PDB2R5P. This is probably an easy one for you to view with that tool that we used earlier. If you want to take a look at this is probably one of the easier ones so far. And this is what the drug is. And this is a human-created drug that kind of just complementarily fits into the scissors and messes it up. And there are better ones. This just happens to be one of the early ones that was characterized by X-ray crystallography. And so that's indinavir. Isn't that cool? And what it basically does, like in a nutshell, is it kind of it's sort of because when the I don't have a good figure for this second, but what is it doing? It's cleaving the protein, right? So what normally happens is the protein sneaks through here, and this is a protease that cleaves at different parts of the protein, right? And it's a pair of scissors, right? And it does it like the protein is a series of amino acids that are linked. And there's a transition state. There's a transition state for the hydrolysis of the protein. So the proteins are little amino acids. I'll probably show this later in a better format. But you have a bunch of amino acids, and the amino acids can be hydrolyzed and cleaved. What this does is this simulates the transition state of peptide cleavage. That makes sense? There's a transition state where the amide is being cleaved. The amide bond is being cleaved. And this is like a transition state inhibitor. So it acts like the transition state, the halfway point of being cleaved. And then this just sits in there and jams it. So the protein gets jammed and nothing can't work, right? So that's pretty cool. This is HIV protease inhibition and sort of the receptor of HIV protease inhibitors. Indinavir is an example. Here's just an alternate image of the same exact thing. This kind of shows it's sort of like a hole. So this is a surface representation. You might be able to do this kind of thing with that other tool also, playing around with the surface rendering. I haven't tried that. Pymol does this very nicely too. But yeah, it just sticks in the hole and it jams the scissors. So this is HIV protease with the HIV protease inhibitor, Indinavir. Okay? I'm going to take a 2-second break. Okay, let's keep going. All right, so this is another crystal structure. Just while we're kind of showing some cool crystal structures of how drugs work, zidovudine, which we've talked about a couple times now, it's also called AZT, right? Zidovudine, AZT. And this is an HIV-1 reverse transcriptase inhibitor. It covalently attaches into a growing strand of viral DNA. It's one of the ways we treat HIV, one of the first ways. And this, this is actually hopefully going to be the model for how we deal with coronavirus. These kind of drugs that mess up the growing viral DNA strands. So we talked about, I think, remdesivir, I think it's called. Something. Yeah, I think it's remdesivir was the one I talked about in class. I think that may be a viable drug here pretty soon. Well, how does this look, and how does it interact with the protein? Well, here is a cool X-ray crystal structure that actually shows the protein is the green thing, HIV reverse transcriptase, the viral protein, and this is the growing viral DNA. This is the growing viral DNA alpha helix. So these are, I'm not showing every atom here, but that's what's happening. And guess what happens when zidovudine, which is AZT, azithromycin, gets incorporated into the growing DNA chain? Well, this happens, and this is actually attached. It's not showing the bond, but it is actually physically covalently attached. And then this is AZT. Oh, I'm sorry, my bad. This is a normal nucleotide, but this is AZT, the purple thing, and that's the azide. Remember, N, and it's terminated, and now the virus can't reproduce its own DNA. That's the way it kind of works. And this is the structure of AZT. Remember this? We showed this earlier in the course. It's really cool, I mean, how this works. The problem with AZT is that it has some sort of toxic effects because it is, it's also incorporating into your DNA. And but certainly for early HIV treatment, this was one of the best ways to do it. It is still used though, and again, with coronavirus, we talked about a slightly different drug. It was this remdesivir thing that had more of an adenosine type thing, which is like a two-ring adenosine base, and there's like a nitrile and there's some other stuff. But that's kind of, and not a azide, but it's just kind of like a wrong nucleotide that would be incorporated into coronavirus DNA. Okay. All right, and here's just a different view of the same thing, which kind of shows how the viral DNA is kind of folded in there, and then you have the AZT. I'm showing it now, zidovudine, as these little orange spheres just kind of attached in there, and it messes it up, and that is kind of how this drug works. All right. Deactinomycin, aka dactinomycin. So this is a DNA intercalating chemotherapeutic drug. Okay, and so what is this drug, this DNA intercalator? And what the DNA intercalators do is they kind of sneak into a DNA alpha helix, and it messes up DNA replication and various things that cancer really depends on. And so here's an example, and this is a DNA alpha helix. This is the drug, dactinomycin, that's kind of snuck in there in a way that there's a planar portion that's really just intercalated, stuck in to the DNA chain. This is the drug, the molecule again. It's a pretty complicated molecule like this, and natural product. but it's a DNA interpolator, and it's used as a chemotherapy drug. Okay, this just also shows the atomic structure of the different DNA atoms and the bases. The nucleotide bases are kind of there. You can't see it too well. It's like flat nucleotide bases, and the drug really sneaks into the DNA structure like that. Okay, it says dactinomycin. All right. Okay, so pharmacodynamics, drug receptor interactions. What are some types of drug receptor interactions? Types of interactions between a drug and a receptor. So thinking at the kind of chemical level, and sort of going back to your gen chem almost, like what sort of how do molecules interact in general? Because a drug is really interacting with just a different kind of molecule, a macromolecule. Okay, what are some of these? Electrostatic, which is one type. That's really like hydrogen bonding and van der Waals, and various dipole type interactions. Those are a couple of the types of drug interactions. We're going to go into a little more depth in this later, but these are a couple of them. And what's some examples of these? Well, one is Lipitor or atorvastatin, which is this molecule, and it has these various hydrogen bonding interactions. So the amide is interacting with looks like a serine. It's a hydrogen bonding interaction. If this is a little unclear, you should check the PDF file. I don't know how it'll look in the video, but yeah, you always have the PDF. But yeah, these are all hydrogen bonding interactions with the receptor, which is HMG-CoA reductase, which is the enzyme that Lipitor deals with. Lipitor, of course, is a cholesterol-lowering medication. Okay, so it is used very commonly for people that have high cholesterol. A lot of old people start taking Lipitor later in life to prevent heart disease. But these are all examples of hydrogen bonding interactions between the drug and the receptor. All right, that's pretty easy to see, right? What about another example is of an electrostatic or ionic interaction. So this is a kind of in the van der Waals territory. So this is not hydrogen bonding as such. There may be a little bit of hydrogen bonding between the oxygens and the nitrogens, but also it's electrostatic. There's a negatively charged part of the drug and interacting with positively charged part of the receptor. So it's an electrostatic or ionic interaction. And this is pivagabine, which is a antidepressant. One of the different types of antidepressants. I don't know how this exactly works as an antidepressant, but it is a good example of a ionic interaction in an active site or between the drug and the receptor. Okay. Zalepion is an example of an ion dipole or a dipole-dipole interaction. So ion dipole in this case is like an ion, which would be like a glutamic acid or an aspartic acid, as an ion. It's negatively charged, and it's interacting with like the delta positive or delta negative part of the drug. So negative, it talks to delta positive, and that's a type of a drug interaction in an active site, right? Negative, delta positive. And then this is a sedative, hypnotic, sort of insomnia type drug. Okay, so looking at the top up there, now we have the nitrile functional group. Nitrile, right? C triple bond N. Nitrile. It's got a delta positive and a delta negative, and in this case, there's kind of like the serine or whatever. It's probably the serine has a delta negative on the oxygen. Delta negative interacts with the delta positive. Okay, and again, follow along in the PDF. Maybe have that open or maybe on your phone or something or tablet, so you can follow this as I'm going through this. Okay, so these were examples of electrostatic hydrogen bonding type interactions, ionic interactions, and maybe dipole ion or ion dipole. These are all a little bit different, right? But they're all ways that drugs interact with their receptors. Hydrophobic interactions. What's that mean? Well, maybe got a greasy drug and a greasy receptor. So it's more like induced dipole type things. where you have, yeah, a greasy drug, greasy receptor. So an example is butambin, which is a local anesthetic. It's part of a gel mixture. I think it is like lidocaine and some other of these local anesthetics. And I don't know if it's over the counter or whatever, but this is a drug in that mixture. And this is an example of a hydrophobic interaction between like the ester part and like it looks like an isoleucine. And this is one of the ways that a drug will bind to a receptor is these kind of interactions. Okay. Covalent. That's different, right? A different kind of drug receptor interaction. The idea of suicide inhibitors or suicide substrates. The big one, and we talked about this earlier in the class, was clavulanic acid, which is a beta-lactamase inhibitor. So it inhibits beta-lactamase. And what's that useful for? Well, with antibiotics, penicillins, and drug-resistant bacteria. Drug-resistant bacteria that, like, we give the bacteria like a penicillin or a cephalosporin, and then the bacteria destroys it. Remember how it worked roughly. So this is clavulanic acid, and so it is a beta-lactam, kind of like the beta-lactam antibiotics or the cephalosporins, and it inhibits bacterial beta-lactamase. And how did it do it? Was serine 70 was an alcohol, serine, right? It attacked the beta-lactam and got covalently attached, right? So serine 70 got covalently attached to the drug. Yeah, that's a different kind of interaction, and that's a covalent interaction, and those are often irreversible or semi-irreversible, so it might eventually get cleaved, but there's, at least for a good period of time, the bacteria and its enzyme is just getting destroyed by this drug, right? It's a good inhibitor of it. I think I mentioned before, suicide inhibitors or suicide substrates. It is a kind of a common thing, and there is a pretty good Wikipedia page on those, and you can see other examples of suicide inhibitors and suicide substrates. Dose-response relationships. Let's talk about dose-response relationships a bit. Okay, so we've talked about this a bit, and the fraction of receptors occupied by a drug is a function of the drug concentration, and we have to measure and quantify this to sort of discuss drug efficacy, right? At drug as we increase the drug concentration, higher fractions of the receptors will become occupied by the drug until all available receptors become bound. Okay. And that's when the drug has the maximal effect. So there's a couple ways we can show that, and that kind of idea of a dose-response relationship. There's kind of a bad way and maybe a better way. The bad way is just you have a linear y-axis, which is percent biological effect, and a linear x-axis, which is the amount of drug. And as you add more drug, you get more biological effect. But it's a little, you need to have a pretty widespread distribution of the drug to see a clear dose response. That's why most people do this sort of semi-log scale version where we keep the y-axis linear. Y-axis is linear, and then the x-axis we titrate in a pretty vastly larger amounts of drug, maybe up to it's usually like micromolar or nanomolar up to micromolar, so larger quantities of drug. Semi-log basically means one axis is logarithmic, the other axis is linear, and in most cases, in most of the cases that I see, people use the semi-log scale dose responses to show dose response. Agonists versus antagonists. We've already discussed this many times. Let's quickly go through it again. Drugs are commonly divided into two basic categories, agonists and antagonists. And we know that agonists are drugs that bind to and activate receptors. Antagonists bind without activating them, and they consequently prevent the binding of other agonists. And it's usually what happens with an antagonist. And it sort of deactivates the receptor or the downstream effects temporarily, often reversibly, usually, not always. Sometimes it's actually going to be more permanent, like clavulanic acid, which would covalently bind to the receptor, right? Okay. If you conceptualize the drug-receptor interactions as kind of a lock and key model, then an agonist is a key that fits into the lock and opens the lock. It activates it, right? Antagonists fit into the lock and jam the mechanism, right? It's kind of like the opposite. Okay. All right. So this kind of shows the same idea. An agonist sort of binds to the receptor. Agonist binds to the receptor, causes some kind of effect, like an activating downstream effect. If you have an agonist and an antagonist, so they kind of compete, and there's something happens, and you get kind of less activation. An antagonist alone binds and then causes no activation. So that's a pretty good cartoon-y diagram of sort of what happens. Okay, so when we consider agonists, there's a couple factors to consider. Potency, which is sort of the amount of drug required to produce an effect of given intensity. That makes sense, right? The amount of drug required to produce an effect of given intensity. And we talked about EC50 and IC50. So we've seen this already. Efficacy is the ability of the drug to produce a maximal response, right? And we're going into agonists right now. I forgot to mention that. So we'll first talk about agonists, and we'll talk about antagonists. Efficacy is the ability of a drug to produce sort of a maximal response. So is it good or not? Is it a good drug or bad drug? Is it efficacious? Differences in drug efficacy are evaluated by comparing differences in the maximal response at high doses or concentration. We'll show that in a figure in a second. Partial agonists are agonists that produce less than a full response when they fully occupy the receptors. In contrast, a full agonist produces a full response. Okay, so we have partial agonists and full agonists. That kind of makes sense too. One sec. Okay, so let's talk a little bit about agonists with different potencies. By different potencies, you might say they have different EC50 values, right? Because EC50 is a measure of potency. And this just shows an example of varying inhibitors with varying EC50s. And the EC50, of course, is the concentration of the drug that gives you it's the 50% point. So it's concentration of the drug that gives you the 50% response point of any individual molecule. In these cases, these are all agonists. Okay, and so clearly some of these are different than others, and what are the what's your analysis of this? Well, compound A appears to have the most potency, right? The most potency, and by having the most potency, it has the lowest EC50 value. Remember, we're not doing IC50. IC50 is for inhibitors or antagonists. These are EC50s, which is for for various things, including agonists. Okay, so anyway, so compound A looks like it may have the greatest, the most potency. So it has compared to the others. Compound B also, its EC50 is a little bit higher. Its EC50 is a little bit higher, and so you could say it's a little less potent. And then compound C, its EC50 is higher also, right? EC50 is higher, and it's a little less potent. And then the worst compound of all is the compound D, which has the highest EC50. And okay, and so it's probably the least potent compound. Okay, what else to say about this? What are the actual EC50s? I mean, you could say that compound A is probably about 0.9 or so EC50. They're in nanomolar. That's the nanomolar scale. And so that you could say that is actually pretty insanely potent. Very rarely are drugs sub-nanomolar, like under 1 nanomolar. Compound B is a little less potent, but it's probably about like 3 nanomolar. So that's also insanely potent. Yeah. And the compound C is maybe 10. It looks like it's dead on at 10 nanomolar, and then compound D is maybe a 50 or so, 70. It's hard to say with the scale. All right, what else to say about these compounds? I think that's pretty much it. Does this necessarily mean that compound A is the best compound, the best drug? I mean, typically we like small EC50s, or if you have an inhibitor or antagonist, you want a small IC50. But that doesn't necessarily mean that A is devoid of side effects or toxicity or something like that. So A may actually be potent against a specific drug target, but it may also have toxic side effects or something. It could very well be that compound D is the best one, if it has a balance of all the different factors that we consider in drugs. Okay, so potency potency is one of the factors we consider, but there's also side effects, toxicity, pharmacokinetics, PK, right? And the A could be rapidly metabolized by your liver, so destroyed. But certainly considering just this variable of percent response, it looks like compound A is the best. Partial agonists, we talked about this a bit. So these are different drugs that have kind of like a different level of percent response, And so considering molecules A, B, and C, and D here, and we're looking at percent response, compound A appears to have the most response at high concentration, at high drug concentration. Compound B is maybe a little less efficacious. Compound C is a little less efficacious. Compound D looks devoid of efficacy, so it's actually not very good at all. One interesting thing is each drug has the same EC50 value, but they all differ in the maximum response. That's often that they produce at high concentrations. That's often the case for partial agonists. Okay, and so if you have a different level of response for each of the drugs, they often have the same EC50. Not always, but in this case they do. And you might say, oh well, wait, do they really have the same EC50? But the thing, because like if I look at compound C, I mean if I look at the y-axis, well, it never actually achieves 100% response anyway. So how do we define percent response, right, for compound C? And how do we say that compound C has the same EC50 as compound A? That doesn't totally make sense. But the thing is that in determining EC50, you would look at the maximal response of each individual compound. Compound A has maximal total response at 100%, and compound B, it looks like it's like 70% maximal, but you would determine EC50 based on the pretty much the height from the baseline to where B is, and that all works out to be the same EC50. Hopefully that makes sense. But the point here is that each drug has the same EC50 value, but differ in the maximal response that they produce. Another point, compound D, is it good or not? Well, it doesn't look very good. It has a very low efficacy. So would you even call it an agonist? I don't know. That would be arguably not an agonist, but is there something else we could call it? You could often call compound D an antagonist, right? It may block the receptor site and produce low efficacy, and you could call that like an antagonistic response. So something like compound D, you might actually relabel as just called an antagonist, I guess. Low efficacious agonists might be considered inhibitors or antagonists. Hopefully that makes sense. Okay, so yeah, we're going to stop here. I'm going to get into antagonists and inhibitors next time and keep going from there. All right, enjoy the video. And ask questions in office hours. I will have a new homework set, but it's probably going to be after spring break just because we're pretty busy right now and getting used to this remote teaching stuff. All right, I'll have another lecture later this week. I'm actually kind of thinking Friday at this point, but as promised, 2 lectures a week. It's just when they arrive may change a little bit. Okay, see you guys. Thanks.